Hello, good morning, good afternoon, good evening, wherever you are in the world. Welcome to No Food Live. I'm your host, Matthew Lang, joined by my co-host, Courtney Riggle. And we're on our 5th episode of the No Food series, where we dive deep into the world of food informatics and the Internet of Food. No Food Live airs right here on the third Thursday of every month, and you'll never miss an episode if you hit subscribe on the YouTube channel.
Hi, everyone. We're so pleased to have you join us today, as Matthew mentioned, for our 5th episode, and we've already covered a series of really diverse topics, which of course we invite you to watch on YouTube if you didn't see them live. Our topic today is actually the second part of a 2-part series on food composition. The first, which aired last month, featuring 3 research scientists, Daniela Barile, Amir Taha, and Selena Wong, who work on the cutting edge of food composition research and connections with human health. Part 2 today will tee off of this fundamental research part and investigate innovations and challenges around curation and management of food composition data. So now that we have this information, how can we use it?
Right. So previous to last episode, we discussed how the semantic web of food, that is ontologies and vocabularies, can enable alignment and interoperability of food systems data up, down, and across food value chains. In this episode, we're going to bring those two worlds together. And what a panel we've got. If you've ever wanted to know anything about building food composition databases, you'd be lucky to talk to any one of our guests today, and we've got 3. We'll be starting with David Wishart, a professor of computer and biological sciences at the University of Alberta, and he's also the director of their Metabolomics Innovation Center, where they have a compiled assortment of molecular databases across the domains of food, drugs, and human metabolites.
Second up today will be Bruce German, a professor of food science at University of California, Davis, or UC Davis, and director of the Foods for Health Institute. Bruce is also lead scientist for the Periodic Table of Food Initiative.
And we also have Naomi Fukugawa, the director of the USDA Beltsville Human Nutrition Center, and one of the main drivers of the USDA's Food Data Central, the most used food composition database in the world.
So let's get started. First up, as we said, we have Dr. David Wishart, a distinguished professor in the departments of biological sciences and computer science at the University of Alberta. He led the Human Metabolome Project, or HMP, a large collaborative project that has identified more than 115,000 metabolites in the human body, research which is all archived on a freely accessible web resource called the Human Metabolome Database, or HMDB. Professor Wishart currently co-directs the Metabolomics Innovation Center, or TMIC, Canada's National Metabolomics Laboratory. Welcome, Professor Wishart. David, we look forward to hearing more about your database efforts, including and especially FoodDB. And quickly before you start, let me remind our live stream audience that you can type your questions into the YouTube comments at any point and we will respond to them live. So David, can you walk us through your work and, relative to food composition and health databases?
Absolutely, Courtney. Thanks very much. I guess I'll show the slides now. Anyways, it's a real treat to be here. I'm going to talk about 4 different areas in the area of food informatics and try to bring together some of the elements of the shows that you previously mentioned, from food composition analysis to ontologies. So I'm going to be talking about food metabolomics, and that's about acquiring data. And so this is just a little different approach to getting quantitative data about what's in our food. And then I'll be talking about those food composition databases that Courtney just mentioned. So that's on the topic of housing data. Then I'll dive in a little bit into food ontologies, but more in the area of chemical ontologies for those components that are in food. So this will help us understand more about the composition of food. And then I'll briefly talk about using machine learning and food data with the idea of actually trying to use all this data that's housing or housed in these databases. So I'll briefly mention this concept of food metabolomics. The idea is that when, as Courtney mentioned, we've been trying to use metabolomics to understand the metabolites that are found in humans, but we can use the same technology to understand what's in blood or urine or tissues in humans to understand what's in our food. And whether it's beginning with the food samples and extracting them, we still use the same approach as we've done with the Human Metabolome Project, using mass spec, NMR, liquid chromatography, and then using some pretty advanced data analytics to identify and quantify hundreds, if not thousands, of compounds at a time from a single sample. We've been working on a variety of kits and systems over the years, some for NMR, some for mass spec. This is an example of one of the kits that's been developed to measure a little over 700 different compounds. Not only identifies, but quantifies with very high precision a whole range of lipids, fatty acids, organic acids, amino acid derivatives, nucleic acids, polyols, and a whole range of other things. Now, these are general classes, but many of them couple or are relevant to things that are important in food taste, aroma, and flavor, as well as their nutrient composition. So this is what's possible, and we've been using these types of kits and assays to start acquiring a whole pile of data about different foods. One area that we've been looking at is analyzing cow's milk, and Bruce will be talking much more about the importance of understanding milk composition. We created this database about 2 years ago where we measured many different samples of milk, but also scanned through the literature, tried to acquire what was known, and then we placed this into the milk composition database. So it has quantitative metabolomic data with the literature-derived values. So right now, we know that there's about 2,300 compounds in milk that have been reported. Some of these would obviously include things like contaminants, herbicides, and pesticides that sometimes make their way. But most of these are relevant to the nutrients that are of relevance to food science. In all of these compounds, we have not only information about their structures, but also their descriptions, as well as calculated properties, spectral properties to help people reference and identify other compounds or related compounds that might be found in other kinds of milk from other mammalian systems. We've been using the same sorts of kits that I just mentioned to start acquiring data about polyphenols. And this is a project that we've been working with a group in France for many years. This is a data resource called Phenol Explorer. And this not only combines experimental work that we've collected, but a lot of literature-derived data from many sources. So compiling these databases takes a long time. It's a lot of work and often involves teams of curators and then software developers to assemble the databases, format them in a way that's presentable, and put them onto the web. The Milk Composition Database, the Phenol Explorer, are all examples of databases that are freely available. Phenol Explorer has been expanded multiple times. I think we're now in version 4. And it has not only information on polyphenols and their plant origins, but also the polyphenol metabolites that can be found in the human body, and then also the effects of cooking and preparation on polyphenols. All of that data, whether it's from the Milk Composition Database, the polyphenols, or other efforts that we've undertaken in our group, is now in this resource called FoodDB. It's the Food Constituent Database. So it has a little over 70,000 compounds that we've identified through literature reviews, our food metabolomics assays, on just about 800 raw or partially processed foods. So this is somewhat different from the USDA's database that Naomi's going to be talking about, as our focus is more on the basic ingredients as opposed to the more complex foods that you might find, so lasagna or pizza. But it is from these foods that these constituent or base foods that we make all of the dishes that are so pleasing and palatable. So this resource has a lot of data on concentration values, a lot of references, because it's been a major literature source effort. But we're also filling in information about health effects and flavor and aroma that are associated with the compounds in the foods. So we have links to the foods, and you can see little pictures of the different food items, as well as the chemicals or metabolites or nutrients that are found in each of those foods. Again, it's an online database structure that you can search by chemical structures, search by food types, text, and even by spectral data. Now, in preparing these databases, we realized it's really challenging to find out the really useful nuggets of information. Either you have to do a lot of reading or spend a lot of time searching through the database. And so with large databases, in order to make them both connected, but more like machine readable, we've been working on ontology. So you've heard already about food ontologies. We're working on a chemical ontology, or a chemical functional ontology, to describe the chemicals that we can find in food or other systems, and to relate them to the biology, biochemistry, medicine, food science, and industrial applications. We've broken it down into physiological effects, their disposition, their roles, and their processes. And in doing so, we're filling out the information in FoodDB and other databases like the Milk Composition Database or the Human Metabolome Database to make them both machine readable and computable. It's been an enormous effort. We've been doing it for almost 5 years now. Thousands of definitions and terms and classifications, and it's still ongoing, but hopefully this summer, most of that ontology will make its way into FoodDB and other data resources. So what can you do when you have essentially ontologies? What can you do when you have quantitative data? What can you do when you have information about flavor or aroma or health effects? Well, you can start using things like machine learning and data mining to make this data useful. And there are examples already where people have used a food database to help with predicting flavor or aroma. We've been using our database to help predict the metabolic fate of when we eat something, what happens to those components and how are they transformed through phase 1 and phase 2. People are also using machine learning with the data that we have on predicting health effects based on the chemistry. And other elements are trying to develop food formulation or recipe generation based on the composition of foods in these data sets and databases. I think what it opens the door to these tools, whether it's the ontologies, whether it's the databases, or whether it's the new methods for acquiring so much information about foods and what they do to our bodies, that you can start thinking about precision nutrition in a new way, about using these data resources, about using machine learning and data analytics to come up with useful prescriptions for exercise, nutrition, supplements, and potentially even medications to improve our health and well-being. So with that, I think I'll wrap it up and leave it to our hosts.
So much, David. Thank you so much. That was really fantastic. And I thought I knew all your resources really well, but I just learned a whole ton of stuff. So maybe real quick, before we move on to our next guest, can you tell us, you mentioned something about, I'm particularly interested in how people can participate with what you're building, either by submitting chemicals or helping to curate the data or ontologies. Do you have that built into your workflows now, or do you have a roadmap for how that's going to happen? What does that look like?
Yeah, no, it's a really good point. And what you're bringing up is one of the challenges in data management. We prepare what are called curated databases. So these are ones that are typically internal. So we have trained curators who go out and collect the data. And that way we can maintain a really high standard. If you go to archival databases, so these are ones where the community deposits information into them. So, databases like GenBank, where sequences are deposited, or the protein database. To create an archival database, it actually requires a lot of resources. And we're just starting to do this now, and we're learning how difficult it is with what's called the Natural Products Database, or NPMRD. And this database eventually is going to house information about 300,000 known natural products. And we've been setting up the curation or essentially the deposition process for the last year. So it will be possible for people to participate in this, but through a different database called the Natural Products MRD database.
Mm-hmm. Okay, great. Thanks. I've got more questions for you, but let's let our other speakers get in here and we'll talk together as a group. That's really fantastic. Now let's shift our focus to our next guest, Professor Bruce German. Also originally a Canadian, but I think he's switched now. Dr. Bruce German is a professor and food chemist in the Department of Food Science and Technology at the University of California, Davis. He also leads the UC Davis Foods for Health Institute, where they research food components for generating novel ingredients that improve human health. Bruce, can you tell us a little bit about your current initiatives and the collaborative network you've developed with members of food and tech industries? And I think you've got some nonprofits and other folks in there as well.
Sure. Delighted. And very happy to be part of this very exciting new program. It's wonderful to share the research we're doing on food to the broader community. So I represent the Foods for Health Institute. Our mandate is simple. In the 21st century, what should we eat and what should we grow? And the approach we're taking is to use a very interesting biological model, lactation. Lactation is this remarkable idea that came very late in evolution, and it's the mother-infant pair. Mothers literally dissolve themselves to make a complete and comprehensive diet for their infant. Everything costs a mother, so if it doesn't profit the mother or infant, the component will be lost to evolution just from the cost. But if anything in milk helps that infant to succeed, to develop, to thrive, it's hard to imagine anything under more positive selective pressure as food. So that's what we've been using. And we've been looking to describe milk as food comprehensively, to look at the composition of all the components, including what has been a very difficult subset, the complex biopolymers. Not the small molecules, the essential nutrients. We're talking about the biopolymers, the proteins, the carbohydrates, the lipids, these molecules that are very large. And up until recently, it really hasn't been possible analytically to accurately capture the structures of these molecules, both in identity and quantity. So that's what we've been taking on, is how do we measure these molecules and then understand what they do. I'll give you two examples of where understanding in precise quantitative terms the structures of biopolymers is leading us to understand how diet works. First, of course, for babies, but actually for all of us in a very precise way. So milk is full of undigestible matter. And our goal is to understand what is it that mothers dissolve themselves to make that's undigestible. Carlito Labriglia, a world-famous analytical chemist, developed analytical platforms so that, in essence, we can measure all of these complex biopolymers in milk. These oligosaccharides are actually polymers of sugars, different sugars in very stereospecific glycosidic bonds. And there are dozens of them in milk. And in fact, there are as quantitatively as much complex oligosaccharides in human breast milk as there is protein. So it's an astonishing, in essence, elaboration of evolution to produce undigestible matter in breast milk. Now that we know what they are, the obvious question, what do they do? So one of the thoughts was, well, if they don't feed the baby, maybe they feed something else. Maybe they feed bacteria. So we enlisted the help of David Mills, world-famous biologist. He studies microbial communities and how do microbial communities change in everything from food, wine, to the human intestine. And we isolated the oligosaccharides and provided them to David to ask a very simple question. Do bacteria grow on human milk oligosaccharides? And the answer was actually rather simple. No, they don't. David tried all sorts of bacteria, and typically bacteria cannot release the sugars from these glycosidic bonds. They can't utilize them as food, and they can't grow on them. But then we found one, perhaps not a surprise, from the intestine of a breastfed infant. And this bacteria, when we analyzed the genome in detail, had, in essence, a whole series of genes encoding enzymes to get access, release all of those sugars, making the sugars available, and this bacteria grows spectacularly well on human milk oligosaccharides. So now we realize that the biopolymers in milk are doing something ingenious. Milk feeds the baby, but it also feeds a very specific bacterium in the baby. In essence, what an ingenious idea. Mothers are recruiting another life form to babysit their baby. Next step was, can we get these in quantity, these oligosaccharides? Daniela Barile, who this program already knows, a chemist at UC Davis in the food program, she studies, in essence, how to isolate complex oligosaccharides from human milk and from other sources, and she was able to develop methods so we could get these materials in quantity, grow the bacteria, and then test, well, in essence, do they provide value to babies? When you do that, it's quite astonishing. So normally, breastfed babies without the right bacteria are full of all sorts of bacteria, enterobacteria, Clostridia, frankly, rather unpleasant bacteria. But if a breastfed baby gets Bifidobacteria infantis, then the entire microbial community changes literally within hours. And that microbial community is dominated by Bifidobacteria infantis. It metabolizes the oligosaccharides. It produces short-chain fatty acids, acetate and lactate as metabolites. Those nourish the baby's intestine. The bacteria pushes out, ostensibly, unpleasant pathogenic bacteria, produces conditions that discourages their growth, and dramatically drops inflammation in the baby. So again, an ingenious strategy that when you get the right bacteria, you have a very protective microbiome, you eliminate most of the deleterious bacteria, and you drop inflammation. So what we've realized is that the complex biopolymers of sugars in human breast milk guide a very specific microbial community in a breastfed baby and protects them from all sorts of, in essence, deleterious risks associated with inappropriate bacteria. We've also discovered that over the past 70 years, this bacterium has been declining in babies due to antibiotic use and infant formula and cesarean section births, and babies have been losing this bacterium. And as that happened, we've seen a variety of health conditions begin to deteriorate. So things like allergy, asthma, autoimmune diseases are increasing because we're losing the appropriate bacteria. So that illustrates that by understanding the complex biopolymers of sugars, you can understand complex functions. And now we're turning to, okay, that's how milk does it. What complex carbohydrates should adults eat? And what bacteria should those stimulate as a community in our intestine? So next to proteins. So we're very interested in the digestion of proteins, how well can babies digest, and in particular, premature infants. A major, in essence, increase in premature birth has led to the dilemma. How do we nourish premature babies? They're premature. They have not fully developed. And our question was simple. Can premature babies digest human breast milk? And especially the proteins. So we, in essence, had to get milk back out of a baby. And Dave Dallas, a brilliant young graduate student, went to Mark Underwood, head of neonatology at the University of California, Davis Med Center, and asked, can we, in essence, study this process? So Mark helped us obtain tiny drops of milk back out of a baby as a function of time. So we could answer the question. The proteins, we know the proteins going into the baby, are they digested into peptides and amino acids in the baby? And I'll confess, I didn't think they could. They're premature. But with very exhaustive new technologies that allow us to measure all the proteins in human milk and all the peptides coming back out of a baby, we could answer that question. And lo and behold, babies are seemingly digesting those proteins beautifully. The babies, in essence, after a certain amount of time, are full of peptides, the products of digestion. So it was clear babies are able to digest the proteins in milk, but how? So by taking the peptides that are released from proteins, sequencing them in high throughput, we could take every peptide and map it back to the proteins that they were generated from. And with that information, we could identify the enzymes, the protease enzymes that release those peptides. Computationally, we're then able to, with Nora Caldi, identify there are 5 enzymes that are releasing the peptides from human milk proteins in the baby. What was interesting is babies don't have those enzymes. Where are the enzymes coming from? Well, lo and behold, they're in the milk. Milk is basically self-digesting. The mother provides the proteins and protease enzymes to help the baby to digest. So what we realized from that is you have to be able to get, in essence, breast milk into premature babies so that they can digest these proteins and, in essence, manage milk from mothers so that those enzymes are still active. But it also teaches us protein digestion is not easy. Babies can't do it very well, and neither can the elderly. So we're working to try and assist in everyone who does not have a good digestive capacity to have the enzymes in food that will help them to digest those proteins. Again, precision in understanding what is in food as biopolymers leads us to how can we improve the nutrition that those components are providing. So from all this, we realize there's a need to do this more broadly. Well, it turns out this also was occurring to the Rockefeller Foundation. And the Rockefeller Foundation was taking advice from David Wishart and his program, from the USDA and their programs, and various labs like ours, and realized that we need to take these modern analytical capabilities to measure the small molecules, the large molecules, the biopolymers, and build literally a periodic table of foods compositionally. What in detail is the composition of every food? And so they've embarked on a bold initiative, the periodic table of foods. And the first goal of that is to select 1,000 foods to analyze completely. These are the first 1,000 celebrity foods. These are not the last foods. These are the first foods whose analysis will inspire the scientists around the world to start adding more and more foods, analyze them and understand. These are the foods that are most successful in nourishing people. These are the foods that are most successful in sustaining the environment, the land, and the water. So can we rediscover the concepts of agriculture for the next century using the complete composition of foods to do that? The second part of that is to understand what are all the things about food that make them interesting. Where are they from? How do they, in essence, grow? How do we process them historically? Who do we feed them to? So all of that, what we call metadata, is being incorporated into the periodic table of foods. Now, the technical platform concept is to provide analytical capabilities for all the classes of biopolymers to every analytical lab in the world who wants to do it. So literally to democratize the analytical capabilities of the 21st century for food analysis everywhere. So building analytical platforms consisting of methods and reagents and kits, models, so that everyone can do this. It is a bold ambition, but we're beginning to see it realized. And then finally, of course, this will all be, in essence, assembled into public databases. What David Wishart has talked about, broaden that concept to include biopolymers and foods literally from everywhere in the world. So this will all make possible a completely different way of imagining the food supply. And we think of this as, at the end, something that's metaphorically analogous to a knowledge-based system that we're all familiar with. Google Maps, right? A database of all the roads and highways and bike paths that is in the public cloud, accessible by your personal device. And the value proposition of Google Maps is simple. Where am I now? Where would I like to be? How do I get there? And Google Maps computes your path. So imagine from a food and health perspective, what is my health now? what would I like my health to be, and what diets will get me there. So computationally, you can see, we build these databases, we understand how food works, and then we provide that as the way people can literally guide their own diets for health aspirations that they have. Thank you.
Wow, Bruce, that's interesting. That's such an exciting initiative, and there's so many different questions we could ask you now. But how about just start now and then come back in the end, and we can pull all these pieces together. With how far along is this initiative? What is happening? What is available? Where are you?
Yeah, great question. And it's just starting. In many respects, we're assembling the final 1,000 foods for what we call our celebrity list. the premier showing. The analytical platforms are being developed right now and should be online within months. And the database structure is being developed and will start coming on board in 2022. So it's a new initiative. It's literally global, and it's using resources from both scientists and agriculturalists from around the world, even taking advantage of the culinary experts around the world. So it's happening in collaboration with actually people on this program. So it's a very exciting time. I would say tune in for outcomes now, literally within months.
Great. We'll look forward to seeing that and hearing more from it later. So excellent. So why don't we shift now to our final guest for today, and then we'll bring everyone back for a discussion at the end. So our final guest today, Dr. Naomi Fukagawa, is director. Are you here? Okay.
I'm here.
USDA Beltsville Human Nutrition Research Center. She's a board certified pediatrician with expertise in nutritional biochemistry and metabolism, and also an expert in the role of diet in aging and chronic diseases. So that full spectrum. Dr. Fukagawa previously was a professor of medicine at University of Vermont and served as president of the American Society for Clinical Nutrition and vice chair of the 2010 USDA HHS Dietary Guidelines Advisory Committee. Dr. Fukagawa, Naomi, the USDA Food Composition Database and the nutritional research has been such a foundational resource for decades. So what new challenges are you responding to, and can you tell us what's currently going on at Food Data Central? What are the prospective innovations we should look for?
Well, thank you, Courtney, for the introduction, and I'm pleased to be able to share with you information about Food Data Central, which I think I should remind many of you that the USDA has actually been publishing information about food composition for more than 100 years, starting with Wilbur Atwater in the 1800s. And right now, for the many of the reasons that you've heard from both David and Bruce, we did feel that we needed to change. And why was change needed? And the change is needed because the food supply has certainly exploded in the last 100 years. There's also an increased need for understanding the relationships or interrelationships between dietary intake and health, and that is greater than ever. In addition, we've had rapid evolution of technology and communication, and there's an increased demand for transparency and easy accessibility to the data, and this has skyrocketed in the last decade or so. Finally, we do know that food systems are global, and they impact not only food security, but also the environment and public health. So therefore, the USDA decided to have a response, which is called Food Data Central. And in this effort, we've tried to centralize food composition data that's been available through the USDA websites, as well as to shift the focus a bit towards research, such that we'd be able to link production of food to products and then to health outcomes. So many of the same things that you've heard from both David and Bruce in the last 20 or 30 minutes. In addition, what's very important for our Food Data Central is trying to address variability in food components. And you notice I say food components. Many for the reasons that Bruce and David had also said, that it's really more than nutrients that appear in food that can have an impact on health and well-being. So we're interested in the variability of components that are inherent, that are affected by the environment, processing methods, and also in food preparation. So this next slide shows you that our new Food Data Central has 5 data sets. Foundation Foods is our newest, and Experimental Foods, bolded here, are our newest data sets. And in Foundation Foods, we are beginning to focus on aspects of foods that are used as ingredients, that are considered staples and or are commodity items. For Experimental Foods, we are hoping or planning to have experimental data which may not necessarily be information about food that's in the food supply, but really to try to relate things to experimental conditions that scientists have developed to try to understand how the environment, cultivar, processing, etc., might impact the component profiles of different foods. Classically, we do have the 3 as previously known to be published for the USDA food composition data system, includes standard reference, which is standard reference legacy, which represents more than 30 years of information about food that was presented in food composition tables at the USDA. And this is what David would probably call archival, and in a sense also includes foods that were prepared or appearing on the marketplace. But as I described earlier, because of the explosion in the number of different foods that we have in the food supply, it became necessary for us to refocus on Foundation Foods and Experimental Foods. Secondly, we have the Food and Nutrient Database for Dietary Studies, which is FNDDS, which is the NHANES survey, what we eat in America, and presenting foods for every 2 years of what people in the US are consuming. And finally, our newest data set, which was released in 2016, is the Global Branded Foods database, which are label data from foods that are on the market that's provided by the food industry and is supported through a public-private partnership. So this would be the home page that you'd see on our new USDA Food Data Central, which, as I said, was released in 2019. And as you can see here, you are able to download the data, get an API key, and we have guides and so forth that can allow you to use this information as in whatever manner you wish to do it. So our focus is really to present at this present time new information on Foundation Foods. So we released a new version in April of 2021, and in this version we have enhanced searching, in which case now you can either look for food by the name of the food, or you can search by components, which would include the different nutrients or components that may be in that food that's of interest to you. And what you can see is that there are different tabs for the 5 data sets that we have available to you, and you can see the number of foods that appear in each of that data type, since each data type is unique and can be used in multiple different ways. In addition, what we've done in 2021 is our first step at trying to begin to incorporate ontology mappings into Food Data Central, which will allow, as we heard earlier, connections between multiple types of data sets and allow investigators as well as people who wish to understand not only the chemical side of what's in food, but also interest in terms of where that food came from, the cultivar or variety of that particular food of interest. So hopefully you'll be excited about going to see our new release of Food Data Central. In addition, I thought I should brag a little with respect to Food Data Central statistics. And namely, we do have what may seem like a small number of foundation foods, but this really represents almost 2,000 samples. In addition, we do have experimental foods, 11 of them at the present time, and more than 300,000 of branded foods. And the survey has 7,000 foods included, and Standard Reference Legacy has about 8,000. So that's quite a lot of data that would be available for multiple uses by different constituents that would be interested in using our data. And we are proud that it is the #1 API for the USDA data sets on data.gov, and we have an average of 3 million calls to our API every month, over 3 million unique users to the FDC website, and interestingly, 43% of our users are actually from outside the US, and the database itself contains information of approximately 6 million nutrients. So where do we see ourselves going in the future? So our short and longer term goals for Food Data Central will be an expansion of experimental foods. We also need to refine our ontology efforts. We hope to increase our website and API functionality, and we get feedback from our users that allow us to try to refine things to make it more user-friendly as well as usable for research and other applications. In addition, you may or may not know about the USDA having a grand challenge for looking at specific agricultural challenges that we face. And so we eventually will be presenting data on the results of grand challenge projects that have occurred, the first of which was focusing on dairy. In addition, we plan to expand it to include information about animal feed, since much of the USDA food composition data system had focused on food for humans, but we also realized that a significant number of agricultural products are developed for animal feed and wellness. And finally, you heard from Bruce about the periodic table of food, which is something that we're hoping to be able to host on Food Data Central after it has been curated and ready for release to the public, because we are a public facing data system. So with that, Matt or Courtney?
Great. Thanks, Naomi. What a valuable, important resource. I just have one quick question for you about Food Data Central before we kind of go into the discussion with everyone. You talk about this new experimental foods data set. So what kind of foods are included in that, and what do you have in there so far?
Well, so far, most of the information and data we have are related to research projects that were conducted by USDA and or USDA-supported scientists. And much of this are already published data in the scientific literature, and it's available as data tables. However, the future is that we would like to be able to entertain the housing of raw data or metadata related to experiments that may not have necessarily impacted the food supply yet, but will give us an inkling as to what the variation and what conditions or cultivars that might influence the nutrient and or other component profiles in food. So that is a data set that's in progress, and we can discuss it a bit more and later in the discussion if people are interested in how you could contribute to it.
So is that okay? But is that like insect protein or formulated foods, or just curious what qualifies as experimental?
Well, usually it's grown or produced under experimental conditions.
Okay, great.
Okay, and initially we have been focusing largely on food that will eventually enter or be related to the food supply. And so oftentimes what's in experimental foods will also appear in a different form under foundation foods. But certainly, if one is interested in insect protein or alternate sources of protein, that certainly could be something that we would eventually include. But it is in progress.
Great. Look forward to seeing where that goes. So Jake, why don't we bring everyone back for our discussion now? And Matthew,
Yeah.
I think you had a question ready to go.
Actually, yeah, this is really great, and I know we are we're running short on time. So, and quite frankly, I want to put a couple of you in the hot seat. So I'm gonna start with you. I'm gonna start with you, Bruce. So you've launched how many businesses now? I think at least 3 that I counted on online related to functional ingredients. And if it's not too much to ask, I think because you would be the business expert here, then where do the business opportunities lie? And what's the value of having this sort of open and public infrastructure, not just for improving health, but actually for a new economy?
Yeah, that's a big question.
Yeah. Well, I mean, you're building the infrastructure, right? And you're talking about building an infrastructure, and you're also are a cutting edge businessman, a founder of companies. So I'm just I think it's fascinating. Our audience is both technical people and scientific people. So where do those intersections lie, and what are those opportunities?
Yeah, sure. And I think I'm speaking for all of the panel, that the world genuinely is faced with some major challenges from the agriculture and food sector. We're going to have to revolutionize what we grow, how we grow it, how we nourish people, because the system in many respects is not the way it should be. It's not sustainable for the environment, land and water, and we're not nourishing people as well as we can. So the most exciting thing about the new tools that both David and Naomi talked about is these tools are literally built for the population. They're by definition public. So we're bringing scientific knowledge and making it accessible. But that gets you most of the way, but now you have to bring it to practice. And scaling science into products and services that help people. We're interested in mothers and babies, and we want to help mothers and babies all around the world as best we can. In some cases, that means disseminating knowledge, but in some cases, it means we have to literally work with the innovation teams at, in our case, UC Davis, to bring those capabilities to practice. But this is a revolution that's literally urgent. We need to change the world's food supply. And to be perfectly blunt, the people on the program are the kinds of people who basically say, no, I know it's going to be difficult. I know everybody else says it can't be done. It just has to be done. And so the things that David has done have literally changed the way people perceive not only what science is doing, but what science could do. And Naomi's program is just the most inspiring thing you can imagine, because it says we literally can change all of these aspects of agriculture and food for everyone's improved health. It's a massive undertaking by any criteria, and most people just, I can't do that. But to just say, no, we're going to do this. It's wonderfully inspiring. And food as a subject is something that's accessible to everyone. I want every young person who's thinking, well, maybe I don't have a career in science, technology, engineering, and math. Yeah, you do. Come into food.
Great. Thank you. That's yet again even more inspiring than you were previously. I don't know how you do it. You just keep geometrically expanding inspiration. So thank you. Dive into a technical question here from one of our previous guests, actually, Damian Dooley. I will put the question up on, and I'm gonna ask this of you, David. Put you in the hot seat a little bit. Damien asks, what efforts are being made within the projects to use externally curated, that is intra-agency vocabularies, like the Obo Foundry ontologies? Naomi put a list of the ontologies that are being used, and those are Obo Foundry ontologies. They interoperate with other ontologies. Can you tell us plans about the ChemFO ontology that you've developed, and any plans you have for integrating that with other publicly available ontologies?
Yeah, no, that's a great question. We're certainly trying our best to integrate with existing public ontologies as we move into particularly the health aspects, trying to use those standard ontologies. We also have to deal with how, when we talk about health effects, food components and food constituents interact with proteins. There's targets, there's transporters. Again, we're trying to use the standard ontologies whenever we have to link with protein data. Again, we're trying to use standard ontologies with the foods. And in other cases, we're finding that we have to kind of go on our own because the stuff we're trying to describe is kind of new, or the current ontologies aren't quite sufficient for what we really want to describe. I mean, in the end, we're trying to create something that appeals to the certain users, whether it's food scientists, but also to consumers or physicians. And so sometimes it's a bit of a mix and match, but absolutely we want to try and use the lingua franca of science.
Right. Well, I'm going to go ahead and extend the invite to the International Food Ontology Working Group, and we can catch up on that later. I think Naomi comes to that meeting once a month, and I think it would be lovely to have a representative from your lab there.
May I? Oops, sorry.
Yeah. Please.
So may I just chime in that, I think one of the things that we really are struggling with is the fact that there are many, many different terms and words that have different definitions for all of the different data sets and ontologies. So having that connection is really a challenge. And our perspective is the fact that we could perhaps at least link our databases because they would be on a common platform, will allow us to probably refine this. And so that's what we're truly struggling with, to try to harmonize vocabulary together with, the health sciences, the agricultural sciences, and the basic sciences at the molecular level.
Well, let's go to a couple more of our audience questions. And I think a couple people are asking this one here that Andy Shriver sent in for Naomi about plans to expand Foundation Foods.
Oh, we definitely wish to expand it. And one thing that I may have neglected to say, that what Foundation Foods consists of is really all analytical data. And so needless to say, that takes time, effort, and money to do all of the analysis for the various components. So we're a bit slow in that what we're trying to do is at least grow from 10 to 12 initially, which is what we released, to more than 30 to 40 new ones a year. And it's slowly building up the database that can then be used in conjunction with FNDDS as well as SR. But it's because of the analytical capacity and the selection of foods that we are facing challenges, because we want to represent variability. Because the food we analyze is not necessarily the food that you eat, nor is it necessarily the final plate that you consume that would be having an effect on your wellness and health. So we are planning to grow.
Right.
Great.
Great. And I can take it. Whoops. We also have a question from Hande, and thanks for the props there, Hande, for the guests. Hande asks you, David, it looks like FoodDB is importing a lot of data from different databases, and you mentioned the curation process. Can you dive into that just a little bit more about what does that look like when you're curating FoodDB and any of your other databases? What's involved in that?
Yeah, I mean it's an arduous process. So if there's data that we can get from other databases that's publicly available, and we consider robust. Obviously, we bring that in, but we do a lot of literature, primary literature search as well to help validate some of that. Of course, we also use our own experimental work to see if we get something that's reasonably consistent as well. So it's a lot of cross-checking, and some of it's manual. It's in terms of sort of the QC process. The idea is to have as many curators cross-checking each other's data. We have a lot of computational tools that use other databases that we've created, which are, in some of those cases, which were very, very thoroughly QC'd. And then using that reference data set that we have from our other databases, it allows us to sort of elevate the quality control of new databases that we create, like FoodDB. So it's a pretty complicated process. I wish I could say it was all written out in a perfectly clear SOP, but it's always iterative because we're always finding new things, new ways, and potentially better ways of doing it.
Awesome.
Great. Thank you. So here's another technical question, and this one, let me put it up here. This is for you, Bruce. Siobhan asked about specifically about the infant breast milk. So does a single dose of B. infantis to breastfed babies adequately populate, or is it a continuous supplemental need? So, how much of this substance?
Yeah, great question, Siobhan. And all indications are, though clinical trials have not pursued all of these aspects, is the genius of evolution is that when Bifidobacteria longum, subspecies infantis, this strain of bacteria that co-evolved with human breast milk, if it's introduced, in essence, once live, then it grows, it multiplies, and within hours, the baby is, in essence, fully inoculated, and the numbers of Bifidobacteria infantis are spectacularly high, and they stay that way. So the bacteria basically is consuming the components of breast milk and continuing to multiply. However, having said that, what we see in multiple babies is this bacterium is very sensitive to antibiotics. And antibiotics, as an example, are relatively ubiquitous in the environment. Typically, not insignificant number of mothers have antibiotics at birth. And so the antibiotics will pass through the milk and knock out infantis. Babies sometimes get antibiotics. So the bacteria stay until in essence, destructive event, typically with antibiotics. And then you have to re-inoculate. And you have to re-inoculate until the antibiotics themselves have gone down. So it's not a simple answer, apologies, but in general, this ingenious idea of evolution is self-perpetuating as long as mothers continue to breastfeed. Breastfeeding is the secret. I don't know how I can encourage more the support publicly and privately for breastfeeding. It really is good for babies. And if infantis gets in there, and away it goes. Once breastfeeding stops and complementary adult foods come in, then the advantage that breast milk provides this bacteria is gone, and it gradually goes away.
Awesome. That's great. And you're getting lots of props in the chat, all three of you. This is fantastic. I want to get back to the database question and ask you, Naomi, in particular. There's a question from Patty James asking whether we distinguish between nutrients that are in raw foods versus cooked foods inside of the databases. And maybe this could be for you as well, David, whether FoodDB categorizes that. What impact do we know processing methods have an impact on food composition. And it seems like there's a lot of variability in processing methods. So how can we capture those kinds of things inside of food composition databases?
Well, I'll answer the question from the FDC perspective, and that
Right.
many of the prepared foods that appear in SR and that appear in FNDDS are related to them being cooked or as eaten or as consumed. The foundation foods, we are starting really with the raw version of most foods, and we'll eventually get to different ways of cooking. But as you know, to try to standardize a cooking method is a challenge in and of itself, and especially if it's part of a mixed dish. So we thought, since the emphasis of Food Data Central, specifically foundation foods, is really to begin to include more analytical data. And these are analytical data that are done on that particular food using vetted analytical methods. And so, in a sense, what one can do is use the information in foundation foods and then create your recipe. And then you could get an estimate of what the profile might be in that particular food that you're having. So it's definitely a shift from what we have previously published in the USDA food composition databases. And especially since we have branded foods, which more or less tells you what's in that package of food that you might purchase in the grocery store.
Right, right. And so when you say as prepared or as eaten, so I'm reminded when I met my wife, she came from Italy, and we talked about boiling or steaming foods. And I'll just use broccoli as an example. We both like broccoli. When I boil broccoli, I boil it till it's bright green. It's still slightly crunchy. When she boils broccoli, it's dark green and not crunchy at all. And there's going to be a huge difference in nutrients. And in her mind, the broccoli that I was eating was full of anti-nutrients. So when you say as eaten or as prepared, what does that mean? And who's as prepared by who?
Well, I guess when I say as prepared or as consumed, it does depend on which of the data types you're looking at within USDA's Food Data Central. But essentially, as you've said, your cooking process really does influence what the profiles are. But more importantly, I think that you don't really eat nutrients. You eat food, and you eat food that's oftentimes a variety on a plate. So therefore, that's really what's going to impact your wellness or whatever physiological outcomes you're having. So, for example, if you look at the importance of myrosinase, which is found in daikon, right, for allowing you to release some of the phytonutrients that are in raw or, some of the brassicas, people through history have learned what combinations can allow them to get the most or the biggest bang out of a food on a plate. And that's what we're trying to shift our emphasis on, is that people should know what's sort of in their ingredient or commodity and things, but then it'll depend on how they plan to prepare it as to what their final profiles will be on their plate. I hope that answers your question.
I think that's fantastic, and I think I look forward to. Courtney's back. To being able to get at these data points just a little bit more, perhaps even with some of these food processing ontologies that we've been developed. Courtney, are you with us? Or you're a little bit choppy. All right.
Well, I need to jump in. Hear me? I'm a little
Barely.
choppy. I guess rural network isn't working.
All right. I'm gonna just.
But I think what a great conversation, and please, everyone.
Yeah. Okay. Courtney, sorry, but I know we're out of time.
If you can hear me, give a thumb.
Yeah.
Okay.
It's
choppy.
Yeah. I'm going to go ahead and close out the show. I'm sorry, this is one more reason why we need rural broadband. I mean, this is the digital divide. We are experiencing it right now. Courtney is on a farm in Eastern Washington, and here we're witnessing the digital divide as we speak. So we need rural broadband so that we can better characterize food as it's grown. Anyway. It's been a great conversation, and we are not just out of time, we are over time. So I hope that our audience will join me in virtually thanking David, Bruce, and Naomi for sharing their work and insights with us today. We look forward to future conversations and collaborations. And before we sign off, we would just like to invite everyone to join us for our next No Food conversation, No Food Nut, around precision nutrition and personalized foods. So reminder that we air on the third Thursday of every month, and in June, that is June 17th. And we will be exploring these topics about how we can use these food composition databases that we've just spoken about to deliver more precise dietary recommendations. Are you back, Courtney? Can you give us a wave?
I can give you a wave. Maybe
All
not
right.
a mic, but a wave.
All right. And don't forget, our episodes are available on the YouTube channel. If you like what you're doing, just hit subscribe. All right. Thanks, everyone. And
Thanks, everybody.
stay in the know.
Bye.
All right.
Yep. See you next month. Bye-bye.
Bye.